Thank you so much, Fabio, and thank you everyone for attending this morning. It is currently 4:15 a.m. here in Vermont, in the United States. For those of you that don't know, my name is Josh Bongard, and I'm a professor of computer science here at the University of Vermont. And what I'd like to talk about over the next 45 minutes or so is carrying on from what Fabio was saying. How do we move forward in the current AI summer, the era of deep learning and large foundation models? What is the role for embodiment in all that? And I think the way forward is to focus on trying to create machines that have a rich inner life, and that's going to be the main theme this morning. What is it about the rich inner lives that organisms have that prepare them to deal with the complexity and challenges of the world around them? And I'm going to use this common theme to explore several research projects that are ongoing in my lab, including evolving robots, Xenobots, and some other things if we have time to get to it. So, of course, these days we can't start a lecture without talking about the intersection between robotics and large foundation models. Here's an example from a few years ago. This is Google's DALL-E, where you can prompt this robot using natural language to perform some embodied task, and it does so, usually very well. And yet, as always in machine learning, we need to face up to the challenge. Of, I see there's a message in chat. Just a moment. Let me see if I can access it. Oh. Oh, okay. All right. That's fine. So as usual, we need to deal with extrapolation, dealing with out of distribution natural language prompts. Like in this case, imagine we prompt the robot to look behind the drawers and assume that that's not in the training data. What does this robot do? It's got no experience to reach perhaps under and in this drawer to find this other packet of cookies in the back there. It's unlikely that this very sophisticated robot would be able to do so. How is it that organisms seem able to obey commands or deal with situations that they've never experienced before? Fabio was just asking if it's snowing here in Vermont. So a common example I give is someone comes from a country that's very close to the equator. They come and visit Vermont and they've never walked on ice before. This happens all the time when we have international students. And most of the time, these young people who are walking on campus and they start to slip on a patch of ice, within about 1/10 of a second, they will be able to send commands to every muscle fiber in their body, and there are trillions of them, to catch themselves and recover slipping on the ice before they slip on the ice. How is this possible? They've never experienced this icy environment before. It seems like one of those miracles that living systems are able to do, and robots are no closer to being able to do than they ever were. The answer, of course, from the large AI companies these days would be, well, you just train it on more data. You train the robot on ice and every other environment you can think of. But of course, that's not quite how life does things. So what is it that's so special about life? What is it that still distinguishes all living organisms from all current robots on the planet? And I would argue it's that organisms have rich inner lives. What you're watching here is a developing organism. You're watching sped up a time lapse of its development. You'll notice a number of cells dividing, and a few seconds back, let me see if I can restart this. You'll notice that some of these cells start to pull inward, and you see a stream of cells streaming into a hole that's being created in the organism. There we go. So these cells, I want you to put yourself in the shoes of these cells. An individual cell is a fantastically complicated machine. It's covered in sensors and actuators. These cells are gaining massive sensor motor experiences. There's a breadth of experiences that they're obtaining. And those cells that were streaming into that forming concavity in the developing organism, those cells are gaining a lot of experience in how to move through narrow apertures, narrow areas. And so when this organism develops, it's going to be able to navigate through narrow apertures and deal with situations it's never experienced in real life because its cells, its tissues, and organs have had very similar experiences inside the body that pre-train this organism for dealing with challenges outside the body. That's the difference. So what we work on in my lab, among other things, is trying to learn what it is about an organism's rich inner lives that pre-trains them for dealing with challenges in the outside world, and how do we create similar machines that also have rich inner lives and can practice inside to deal with challenges outside. Okay, so here's my cartoon to try and summarize this. As we know, for organisms and machines, the outside world throws lots of novelty at us, new situations that are far outside what our bodies as a whole have experienced. Organisms deal with this by through development and by passively and actively developing and carrying out and simulating physically a lot of complex dynamics inside, which I would argue pre-trains them for dealing with challenges outside. Okay. All right, so let's look at a few examples. One way we can approach this in robotics is to try and create machines that already have rich inner lives out of the box, so to speak, to begin with. So here's an example of a machine that clearly has a rich inner life. You can see that this machine has a large number of small components. This is a Xenobot. It is a robot made only from living materials. It's made only from cells. And Xenobots are made only from frog cells. You're looking at a Xenobot that's made from about 2,000 frog skin cells. These particular cells are taken from a particular frog called Xenopus laevis, thus the name Xenobot. Okay. You can tell that it has a rich inner life because in this case, we're doing some calcium signaling. We're watching flashes of calcium inside the individual cells. And it looks, in this video at least to me, that these flashes are kind of random. It doesn't look like there's it doesn't actually look like there's a rich inner life. It just looks like a whole bunch of things that have been welded together, a whole bunch of components that have been welded together, and they're all doing their own thing. That being said, you will see that when there's an unexpected event, which is this Xenobot becomes punctured with a very small needle, which you'll see in a moment. After this puncture event, there's the puncture event. After this puncture event, you'll see there's a large global flash. You'll also see cells migrating into the wound where the Xenobot was punctured. It turns out that all of these flashes are not random, and this is the rich inner life. These cells are coordinating and communicating amongst themselves. And we found this out and reported this actually in a recent paper that we published this year. This was a collaboration between my lab and the Michael Levin's lab at Tufts University. It turns out that if you take a Xenobot in the upper left and you plot the concentration of calcium in each individual cell in that video, you get some time series data, which you see in the top center. You can then plot the covariance matrix of this time series data, which is what you see in the upper right. So we have a matrix here where every element in that matrix, the element ij, for example, reports the amount of covariance between cell i's calcium dynamics and cell j's calcium dynamics. And you'll notice that there are large red blocks. So these are areas of high covariance. These are different cells in different places inside the Xenobot that are co-varying together. If you observe any cell I, you can make a very good prediction about what the calcium concentration in cell J will be. So this is not random flashing. There is some coordination and covariation among, in this particular Xenobot, 4 big blocks of cells. Turns out if you take a person and put them in a functional magnetic resonance imaging device, an fMRI, and you record activity in different brain regions, you also get obviously time series data like you see in the bottom center. And if you then look for covariation between different brain regions, you also get regions that covary together. So in this example you see in the bottom right, for this particular person's brain at this particular point in time, there were three different parts of the brain that there were different regions of the brain that were covarying together, and there were three of those covarying regions. My very talented postdoc, Thomas Varley, who led this work, found that the nature of this covariation inside a xenobot is very, very similar to the nature of covariation among different parts of a human brain. Quite an interesting result for lots of reasons. If you want to conclude from that result that xenobots are thinking or that xenobots are conscious, you're free to do so. What's also interesting about this is it suggests that the complex dynamics that we often point to in the brain and say, that's thinking, those complex dynamics show up, or very similar dynamics show up in other parts of the body, in non-neural tissue, in this case, in a xenobot. And I think this is another piece of the puzzle in understanding how the body shapes the way we think. Okay, so let's just go back to the beginning of Xenobots, just to lay the foundation so we know what we're talking about. Back in 2019, just before this 2020 publication, my team teamed up with the Levin Lab at Tufts University, and at that time, Doug Blackiston, the microsurgeon working in the Levin Lab, told our group that he could extract two types of cells from very early frog embryo. He could extract frog skin cells and frog heart muscle cells. Frog heart muscle cells will spontaneously get larger and smaller, which is good news for you, because if you could put a whole bunch of human heart cells together, all those heart cells get bigger together and get smaller together and causes your heart to act like a pump. So in this very first xenobot experiment, we thought about using these frog heart cells as the actuators or the pistons that are going to drive our xenobot. My PhD student at the time, Sam Kriegman, then created a biophysical model, and you can see that biophysical model simulating three possible xenobots. We've taken the two building blocks we have available, frog skin cells, shown as blue cubes, and frog heart muscle cells, shown as red-green cubes. And we now have a genetic algorithm that's put these two building blocks together in random ways, and it's created three random configurations of these two building blocks, and you see those three random configurations here. The genetic algorithm also created 97 other simulated xenobots, and it simulated these 100 xenobots one after the other. And the genetic algorithm assigned a score to each one of them according to how well that simulated xenobot did at the task we wanted it to perform, which in this case was just to walk from left to right along the bottom of the simulated Petri dish, which is shown in white in the videos. Not surprisingly, any random combination of these two types of simulated cells does not produce left to right motion. But if you look very carefully, you'll notice that the bottom one moves a tiny bit to the left more than the upper two xenobots. This is my poor camera work. The camera should actually have been on the opposite side, so you'll have to mentally rotate the xenobot at the bottom. That xenobot at the bottom moves a tiny bit further to the right than the upper two xenobots. It gets a slightly higher score. So the genetic algorithm in this situation deleted the upper two xenobots, the ones with lower scores, and made randomly modified copies of the lower xenobot. So let me show you what I mean by randomly modified copies. I apologize for the color scheme here. The pink and the purple corresponds to skin and heart muscle cells. And you're watching evolution sped up in this case. Every time there's a change in one of these Xenobots, that's that Xenobot producing it's producing a new offspring. It makes a copy, and then during that copy, there's some mistakes. Either some cells are added or removed, or a skin cell is turned into a heart muscle cell, or a heart muscle cell is turned into a skin cell. And just going back for a moment, all of those randomly modified copies are the offspring of that higher scoring individual, and those mutants or those randomly modified copies replace or overwrite the lower scoring Xenobots. So we ran this genetic algorithm, and genetic algorithms have been around since the 1970s, a very, very old and trustworthy algorithm. We repeated this process for about 2 weeks on the supercomputer here at Vermont. And at the end of those 2 weeks, the supercomputer sent us back this video. So you can see that the Xenobot does exactly what we wanted it to do, which is to walk from left to right along the bottom of the Petri dish. You'll notice a couple of interesting things about this Xenobot design. There is skin cells on the top and heart muscle cells on the bottom. You'll notice that the heart muscle cells are not coordinated at all. I mentioned that in Xenobots there tends to be a little bit of spontaneous coordination among the cells, but it's hard to predict where and when that coordination will occur. So we made things hard on our genetic algorithm by not allowing coordination. We just have desynchronized heart muscle cells. They're increasing and decreasing in volume at random phase offsets relative to one another. But despite that random desynchronized behavior, we get relatively coordinated and non-random motility or movement. This again demonstrates one of the things that we all kind of know by now about AI, is that it can often generate something that's better than something that could be generated by a human expert. Very hard for a human engineer to design a reliable machine out of unreliable parts. Okay, so this is the end of stage 1 of the experiment, which is the AI part, getting the AI to automatically design a Xenobot for us. Phase 2, which I'll show you in a moment, is the manufacture of this automatically designed construct. So I'm not going to show you all of how this works, but I'm showing you a few videos that were collected by Doug Blackiston, the microsurgeon. We're now looking down through the microscope with Doug. And at this point in the construction process, he's liberated about 1,000 skin cells from early frog embryo. He's injected those individual cells here into the bottom of a Petri dish, and that black circle that you see, that's a small indentation in the bottom of the Petri dish, and those cells settle into that indentation and form a shallow carpet, a small thin film or layer. Doug then puts more skin cells on top, more heart muscle cells on top, heart on top, skin on top, and sort of builds up the cell distribution according to this blueprint. Sorry, I misspoke. Puts down heart muscle cells first, then a little bit more heart muscle cells, skin, 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 to get this cell distribution just about right. After he's done that, those cells are adhesive. They tend to stick to one another, and you get this sort of blob that you see here. And in this last stage of construction, Doug has reached in with very, very small microsurgical devices and is scraping away the unwanted material to try and match the 3D geometry that was discovered by the genetic algorithm. So in a nutshell, or basically in this construction phase, Doug is approximating the cell distribution and approximating the shape of the AI-generated Xenobot. And then the very, very last stage in this experiment is Doug is going to take this Xenobot, put it right side up on the bottom of the Petri dish. Sorry. And you'll see in a moment that the physical Xenobot moves more or less like the AI predicted that it would, and it has more or less the right shape. It's not perfect, and that's actually good news for us. It means when we try and cross from simulation to reality, reality doesn't have to match simulation perfectly. It can be an approximation, and we still get the desired behavior, which is about a millimeter-sized Xenobot walking along the bottom of a petri dish. And I realized I skipped over a slide. I didn't give you the context of the size of a Xenobot. So hopefully you can see this on your screen. Way over here on the left, there is a very, very small black speck. That's a Xenobot. So if you were to look into a petri dish with the unaided eye, not through a microscope, you'd see something that's about the size of a poppy seed that's very slowly moving around. So that was 2020. That was our demonstration that we could combine synthetic biology and AI to create whole new kinds of organisms that haven't existed on Earth before, at least as far as we know. It's also a demonstration that we don't necessarily need to build robots from technological components. We can build them from biological components. And that obviously has many advantages, and one of them, as I mentioned before, is that this Xenobot, while it's walking along the bottom of the petri dish, exhibiting external behavior, there is also rich inner dynamics going on. And one of the goals for my lab at the moment is to try and understand how those rich inner dynamics help and enable, in this case, a Xenobot to do what it does and adapt to changes in its environment. Okay, it's also fun to think about potential applications for Xenobots. Xenobots are still a relatively new technology, so we're working on applications. Here's a Xenobot swarm, and like the robot vacuum cleaner Roomba, Xenobots will just, by chance, as they move around, push very small bits of material in the petri dish into piles. Possibly that could help us in future by cleaning up microplastics in waterways, or possibly cleaning up cancer cells in our bloodstream. I've been talking about Xenobots today. The Levin Lab has also now managed to create anthrobots. These are biological robots built not from frog cells, but built from human cells, which obviously opens up potential medical applications. Some other interesting things we've managed to get Xenobots to do is we can genetically engineer them so that they fluoresce different colors, or they glow different colors. That's useful in trying to design Xenobots to do simple robot behaviors like go, see, remember, come back, and tell. There's thousands of applications where you'd like a robot to go, see, remember, come back, and tell. Here's an example of that. In this case, we have Xenobots that fluoresce green by default. That's just what they do. If we take these particularly genetically engineered Xenobots and put them in a petri dish and they start to move around in the petri dish, if they come into contact with blue light, which we've designated as something that's of interest, that's the thing we want them to go, see, and remember. If they come into contact with blue light, they switch to fluorescing or glowing red, and they will continue to fluoresce red even if they move outside the beam of blue light. And if then at the end of this experiment we collect back all the Xenobots in the dish, or we collect them from some collection area in the dish, and we count the ratio of green to red Xenobots, we can estimate the fraction of them that have been in contact with this substance of interest. That's the ones that have gone, seen, remembered. They've come back and they've told us that they've been in the presence of red light. So the current work between my lab and the Levin Lab and the Blackiston Lab is trying to recapitulate or redo all of the basics of robotics. What is basic sensing? What is basic internal processing? What is basic actuation in biological robots? And how do we then put together sense, think, act loops into more complicated sense, think, act loops inside Xenobots to achieve more and more complex and useful behavior? We're not at useful behavior yet, but we hope to get there in a few years' time. Okay, I'm going to switch now away from biological robots back to traditional robots, or relatively traditional robots made from technological components rather than biological components. I mentioned that our goal is to make machines with rich inner lives. So one way to do that is to create soft robots. We're able, as a community now, the soft robotics community, to make robots out of increasingly sophisticated behavior, out of increasingly sophisticated materials, which opens up the opportunity for them to have rich internal dynamics, and potentially in the future then exploit or use those rich inner dynamics to prepare them for the challenges of the external world. Soft robotics is still a relatively new field, but making good progress. One of the challenges of soft robotics from the simulation side is obviously it's very difficult to simulate these robots. Here's one simulation we created of some soft robots for for Rebecca Kramer-Bottiglio's lab at Yale University. The simulation in the top, as you can imagine, is very, very computationally expensive. It takes a long time to run just this one simulation, which makes it difficult to evolve the designs for soft robots because we take this computationally expensive simulation and wrap it in a genetic algorithm, and that genetic algorithm wants to try out thousands or millions of simulated soft robots. That becomes an extremely computationally intensive task. So in recent years, we've been moving away from evolutionary algorithms, which rely on random trial and error, and moving to gradient descent methods, which are capable of optimizing designs non-randomly. You can guarantee under certain conditions that a gradient descent optimization method will always make a better robot the next time. And let me show you how that works. This paper that I'm going to talk about now, this was a collaboration between myself and my former PhD student, Sam Kriegman, who's now a professor at Northwestern University, and also a collaboration with Daniela Roos, the head of the AI lab at MIT. So in this paper, we demonstrated the bringing together of robot design with gradient descent. What you're going to see in a moment is gradient descent optimizing the body and brain of a robot non-randomly. It means it becomes very computationally efficient. And in this particular experiment, we managed to take what would have usually taken us several weeks on a supercomputer to design a robot. It's now possible to do it within 30 seconds on your smartphone. And at the end of our lecture today, I'm going to give you an opportunity to actually do this on your phone in a moment. But let me explain what's going on under the hood. Let's take a very simple simulated robot, about as simple as you can get. This is a Breitenberg vehicle. I'm not sure if you talked about Valentino Breitenberg's vehicles in this course, but if you haven't, don't worry, they're quite simple. This particular Breitenberg vehicle, as you see here, it's got two wheels at the back and two light sensors on the front. And the left light sensor is attached to the right wheel, and the right light sensor is attached to the left wheel. We have two wires or two synapses in our neural network controller. Very, very simple. If you imagine that there was a light source ahead and to the left of the robot, like you see here, obviously there would be more light falling on the left light sensor and less light falling on the right light sensor. And imagine that we'd like to design or tune the body and brain of this robot so that it turns towards the light source. We want it to exhibit the behavior of phototaxis. Imagine that's what we want, shown by the black arrow here. Let's imagine we just glue these sensors to the robot and we set random weights for the two synapses that connect the light sensors to the wheels. And in this random construction of this Breitenberg vehicle, it doesn't do what we want. It moves up and to the right, away from the light. So it's not doing what we want. So in order to start the robot design process, we compute behavioral loss represented by L here, which is basically just the area between what we wanted and what we got. And we're now going to use the backpropagation of behavioral loss. We're going to take this loss in behavioral efficiency, and we're going to propagate it back through the body and brain of the robot. And as we do, gradient descent is going to slightly change bits of the robot's body and brain to reduce this behavioral loss. Okay, here we go. So the first thing we can do is we can compute, or the gradient descent method in simulation can compute how loss varies as a function of omega here. And omega represents the radial velocity of the wheel, how fast or how slow the wheel is moving. So this particular gradient here, dl over d omega, says as omega changes like this is how loss changes. So if we have that gradient, we know that if we change omega like this, L will come down. If we change omega like that, L will go up. Obviously, we don't want L to go up. We want loss to come down. So we can take this gradient and we can apply it directly to the wheel. We can tell the wheel, you should turn like this instead. But that doesn't make sense in our physics engine, because the turning of the wheel is actually dictated by the strength of the synapse that's arriving at that wheel. So we don't apply that gradient. We keep backpropping. We keep going further back into the body and brain of the robot. And as I just mentioned, the turning of the omega itself is dictated by theta, the weight of the synapse that arrives at that wheel. So using the chain rule, we can work backwards and we could obtain this gradient, which is, this gradient tells us how behavioral loss changes as a function of theta, the weight of the synapse. We can then apply this gradient so that we're confident that we've changed theta in a way that we know L will go down the next time we simulate this robot. So this gradient we do apply, dl over d theta, but we're not, so we've made a very small change to the brain of the robot. We're following just one gradient back through the body and brain of the robot, but you probably know from the traditional backpropagation of error in neural networks that these gradients are propagating backwards through a neural network in parallel. They all go together. So there are also some gradients that are flowing backwards from the lower left wheel up to the upper right sensor. Okay, my apologies, but I can't seem to see chat for some reason. So there may be some questions, and I'm happy to take them when I finish up in a few minutes. So we've got two gradients flowing from the two wheels to up backwards to the two sensors. We've applied one gradient, dl over d theta here, to improve behavioral loss by tuning that right-hand synapse. And we can further continue back to the left sensor, because the spinning of the right wheel is a function not just of the weight of the synapse, but also the horizontal position of the left sensor, which I'm going to represent by X here. So let me just go back and forth to show you how this works. In this moment, where the light source is in front and to the left of the robot, at this horizontal position, there's more light falling on the light sensor, and at this position, at this horizontal position, there's slightly less light falling on the left light sensor. So in this case, this particular gradient, dl over dx, is telling us how to change the horizontal position of the sensor. It's dictating a change to the body. And that gradient tells us that if we, for example, move the light sensor a little bit to the right, the next time we simulate this robot, loss will go down even further. So in the work that we reported in 2023, we showed how gradient descent can non-randomly change bits of the body and brain of the robot, so that now when we simulate the robot the second time, it's going to do a slightly better job of what we wanted it to do. Loss has come down. And now that we've simulated the robot a second time, we can go back and recalculate all the loss gradients, make, again, small changes to the body and brain of the robot, bring loss down again, and by alternating back and forth between these forward and backward passes, we can optimize the body and brain of the robot to do what we want in a computationally efficient manner. Okay, so we didn't do this with simulated Bratenberg robots. We did this with simulated soft robots. And I see that I'm running out of time, so I'm not going to talk in detail about how this soft robot works. But what you are seeing here is the initial random robot. We took a block of soft material, we punched a bunch of random holes through it, and the pink that you see, these are hollow cavities inside the robot, and we attach a hose to these hollow areas, and we can pull air out of these holes or push air into these pink holes and make these pink holes smaller or make these pink holes larger. And that's the actuation of this robot. It's pneumatically actuated. So you can see we've punched random holes and we've created random internal pink cavities inside the robot. And when we push and pull simulated air out of this random robot very quickly, not surprisingly, it doesn't go anywhere. We compute the behavioral loss of this robot, which is the difference between how it moves and what we want, which is that it moves to the right. We do one forward and backward pass, which you can see in the little GIF in the bottom left. And that backward pass is dictating how to move the holes in the robot and how to change the internal pink cavities. Once we simulate those small changes, we get a robot that does this, which you can see moves a little bit further to the right. So at this point, we've done just 2 simulations compared to the hundreds or thousands or millions of simulations we would need to do with a genetic algorithm. Here's the situation after 4 forward and backward passes. And here's the result after 10 forward and back passes. You can see that the gradient descent has non-randomly optimized the body. And the result in this case is actually the appearance of kind of what look like legs and actuation patterned along the anterior or the front part of the legs. So now we can automatically design robots very quickly. And these robots have arguably a bit of a rich inner life. They have a lot of internal hollow cavities, and they have, or at least they used to have, a complex geometry. These robots are not yet actively practicing things inside to pre-train or prepare them for surprises outside. That's what we're working on at the moment, and I hope next year, the year after, to tell you more about that. Okay, we only have a few minutes. Oh, and of course, I should tell you that we, of course, built this in reality. Okay, we have only a few minutes left, so I want to give you a chance to try this yourself. So if you get out your phones and direct the web browser in your phone to bit.ly /3RKEZ3A, it should bring you to a Google Colab web page. I'll give you a moment to type that in. It's a bit of an eye test here, a combination of uppercase and lowercase letters. Hopefully some of you have managed to get there. I'll keep the URL up there for you. If you on your phone have successfully arrived at the Google Colab page, you'll see 3 horizontal bars at the top left. Please tap those 3 horizontal bars. That'll drop down a menu for you. Please tap on Runtime in that drop-down menu. That'll bring up a submenu, and that submenu tap Run All, and that will run all the code inside that Colab notebook on one of Google's data centers. And now wait a minute. So your code is now running, and let me tell you about what your code is doing. Your code will first of all generate a random robot like the one that you see in the upper half of this video. In this case, we're dealing with a spring mass robot. And gradient descent during this minute will now go to work on your random spring mass robot. And hopefully, that gradient descent method will do a 10 forward and backward passes. And when I ran this on my phone a couple years ago now, gradient descent managed to slightly move the gray mass points and slightly change the strength of some of the black springs and slightly change how the red blue springs actuate. And by making those body brain changes, gradient descent on my phone after about a minute was able to design to optimize this random robot into a efficacious robot, a robot that's doing what we want it to do, which, as always in our case, is moving from left to right. Hopefully some of you receive such a video. Depending on your phone, the video will either show up in Colab or it'll be downloaded into the gallery on your phone. And please keep this video as a souvenir from our time together today. And we have now, as a group, I'm not sure how many people are online, we've all now collectively optimized a morphologically diverse swarm. We've all made robots with unique bodies and brains. So if you want to show these off and see what else your fellow students have made, if you post the video to whatever your favorite social media is and add the hashtag AI built bots. When this lecture finishes, if you get online on social media and you search for you search for the hashtag AI built bots, hopefully you'll see some of the videos from your fellow students. Last thing I want to say is I think heterogeneous swarms is the future for robotics. We face a lot of global challenges. One of them is cyber attacks. All of the technology on the planet at the moment, it's more or less the same inside. We have 3 operating systems running on the planet, and in terms of autonomous vehicles, they're all running more or less the same foundation model. This is very good news for the bad guys, because if they can find a way to bring down one autonomous vehicle, that piece of software will most likely bring down a whole bunch of other autonomous vehicles. So one of my hopes for robotics in the future is that we create robots like snowflakes. No 2 robots are the same. They each have unique bodies and brains. They each have unique ways in which they do whatever we want them to do, which in this case is to move from left to right. This makes things very difficult for the bad guys because no one antagonistic attack can bring down the whole swarm. They do not share a common Achilles heel. Okay, I'm going to finish there and thank all of my lab members on the left and all of my faculty colleagues on the right, and also NSF, DARPA, and the University of Vermont for funding all of this work. Okay, thanks very much, and I'm now happy to take a few questions if there's time. Okay. Yes. Okay, I'll do them in order here. The organism from your example is evolving its morphology toward a given architecture that is somehow pre-known. Good question. Sometimes we know what the answer is. So in the very simple case of the Bratenberg vehicle, absolutely, you can sit down with pen and paper and design or set the weights for a Bratenberg vehicle so that it will phototact. That is not possible when you get to any robots that are more complicated than a Bratenberg vehicle. It is extremely difficult to solve the inverse problem, to take some task you'd like the robot to do and work backwards to what is the optimal robot that'll perform that task. That's why my lab does what it does, which is trying to automate the design of robots. Very, very difficult. Next question from Vladimir. The frog embryo. Yeah, sorry, I'm sorry. In the frog embryo case, there's no, of course, the embryo is developing and the embryo is a product of evolution. But when we take cells from that embryo, there's no more evolution and development. We stick those cells together and they more or less do what we want. That's not quite true. The cells are still doing stuff. Presumably, they're trying to fulfill their role in the developmental program of the frog. So they're certainly not passive. They're trying to do things, but we certainly don't know, what it is they're trying to do. We're trying to control that. And in future Xenobot work, we're trying to exploit that. We're trying to build on top of what the cells want to do. Hopefully that answered your question. If not, please feel free to reframe it and type it into chat. Okay, great. Next question from Vladimir. How long can Xenobots operate for? By default, they quote unquote live for about 7 to 10 days. Remember, these are cells from early frog embryo, so they've absorbed some egg yolk from their mother inside the individual cells, and the individual cells are digesting those yolk platelets, and they sort of run out of fuel after about 7 to 10 days. Although Doug Blackiston has now figured out how to get Xenobots to live for much longer than 7 to 10 days, that work hasn't been published yet, so I'm afraid you'll have to stay tuned. Can they operate outside the Petri dish in some harsher conditions? The answer is yes. Obviously, we have to be very careful about that. There's very careful regulation around unleashing synthetic biology constructs into the wild. But we do know, working with some partners who know how to do this very carefully, that yes, they will survive in basically freshwater environments where you find frogs, which is a lot of places. We call these Xenobots, they've got other names, but they are also in a way, genetically unmodified frog material. So turns out there's quite a range of natural environments that they can survive in for about 7 to 10 days. Muslim A says, I have 2 simple questions. What actuation mechanism do you use in these Xenobots? The ones I showed you today, the actuation mechanism is the frog heart cell that gets bigger and smaller. Deforms the body of the Xenobot as you saw, which causes it to move. We have other experiments in which the Xenobots grow small cilia. These are subcellular hairs. They're very, very small. And these cilia grow on the surface of the cells on the surface of the bot. So imagine a bot that's about this big and it's got a whole bunch of hairs on its outer surface, and these hairs are all beating against the fresh water, and they cause the Xenobot to swim through the water. Imagine a whole bunch of small human rowers inside a boat. That's basically what the cilia are doing. So we can make Xenobots that walk using heart muscle, and we can make Xenobots that swim using cilia. What simulation software do you use? We create our own. It's built on top of PyBullet. But if you want to sort of play around with the simulations I showed today, you can do so by following our online course. And I'll type in the URL for that course. It's called Ludobots, and the Ludobots online course will walk you through how to code up some of the simulations you saw today. Let's see. Lucas asks, did you already publish a paper about gradient descent? Yes, that's the 2023 paper. That was Kriegman, Roos, and Bongard 2023. So if you put those keywords into Google Scholar, you'll find the paper about gradient descent. I'd like to learn more about it. Great. Okay. There's a question, Xenobots, biobots, chemical robots, what are the relations between each group of them? That's a great question. I don't think anybody knows. This is sort of, I think, the beginning of this new era in robotics. So we're getting better. We as a community of roboticists, we're getting better at building robots out of all sorts of materials and understanding the advantages and disadvantages of this material and that material. Unknown at the moment, in my opinion. So hopefully you're diving in and you'll help us build a better map and sort of taxonomy of how to build robots out of a broad range of materials. Are there any successful applications of Xenobots. Not yet, but we're working on it. And we do have a spin off company that just completed its seed round of funding this past year. So we are actively working on applications. If you want to learn more about that, the company's called Fauna Systems, as in flora and fauna. So you can check it out. And we're not quite yet ready to roll out practical applications yet, but they're coming. They're coming. Okay, I see I'm a little bit over time, so I appreciate everyone's patience. Oh, great to hear it. Great to hear it. Well, thank you so much, everyone. By all means, Fabio, by all means. Okay. Yeah, for those who don't know, I'm Rolf's former student, and he asked me as a favor 21 years ago, would you mind giving a lecture at 4 o'clock in the morning your time? And I said yes, and I'm still doing it. So there you go.